them until the break. This is just a very quick presentation for each of the posters. At the JKI for vine cultivation, we wanted to identify new resistances against downy mildew, characterize them genetically, and make them available for grape cultivation. We wanted to make sure that when new varieties are bred, that various resistance with different defense mechanisms are brought together in a new kiwi variety, because this will lead to more sustainable resistance. In the first part, we used viticulture as the new resistance against downy mildew, and we examined a cross population, and this was on. We mapped these populations genetically, and were able to identify a new strong locus of resistance on chromosome 14. And the second part, we're interested in the hair on the lower part of the leaf, underneath the leaf, which is a physical barrier against infection with downy mildew. And these physical barriers make it very improbable for the pathogen to be able to learn to overcome resistance. And so the feature layer of hair underneath the leaf is not used in cultivation, but it could actually help as a first barrier to overcoming resistance. We concentrated on Vidus brusca, the North American variety, which has a very dense layer of hair underneath the leaf, and examined the different resistances and 3 different chromosomes displayed loci that are responsible for resistance. We also used AI that we developed for the project, which allowed us quick and objective assessment of the hair based on photos of leaf discs. So all of the features are to be characterized and examined further, and we also want to find the genetic markers for them. And in the next step, we want to use them in cultivation in order to be able to cross these new resistance against downy mildew in new kiwi varieties. If you have any more questions, we'll be happy to answer them at the booth.
Sorry about that. So, but to answer the question of the audience, so we have the solution. So the presentation of Simon shows that there is already fungus adapting to the new peewee. And the solution is to combine several resistance into the new peewee. So the poster show on the right sides the rich vitis diversity present in the collection of the Hochschule Geisenheim University, which could be used to increase the low diversity present of resistance present in the red peewee and white peewee produced in Germany. So 60% of the peewee produced in Germany have only one known RPV marker or RPV resistance, and 3/4 of them are the RPV 3.1, 3.2, 3.3, which is quite weak resistance. So in order to increase the diversity of resistance in the new peewee, we test 74 accessions representing 17 different vitis species from Asia, Europe, Central and North America during 4 summers. So it's representing 35,000 leaf discs inoculated and also scored visually and with using artificial intelligence. And we also check 30 different genetic markers linked to 15 known RPV resistance. What we are observing is that we have several resistance, like resistance RPV1, RPV8, RPV6, RPV14, which are present in Geisenheim but absent in the peewee, in the German peewee, and which could be used to increase the diversity of resistance in the German peewee and solve this problem of adaptation of the fungus. But we have also some accessions. It's not so nice on the screen, but you should go on the poster. We have also a lot of accessions which are showing very clean resistance, very immune, so without any fungus on it. And we have a lot of them, so which could be used for future breeding program. Thank you.
Perfekt. Gut, ja, ich präsentiere gleich das nächste Poster ebenfalls.
Well, this is the next poster, also to your right. The origin and reduction of phosphonate residues in the vineyard. So phosphonate was admitted until 2013, and it has the potential to reduce the use of copper, and particularly in the to tone down the more extreme years and reduce the copper load. And so the question was how to do the analytics, how to see the origin of phosphonates, and how to reduce phosphonates. So the analytics was done relatively quickly. This is a new method in which each compartment of the vineyard of the vine can be examined separately, and particularly we had some residues, and this led us to the next question, which is the origin in organic wines, which should not contain any of those residues. So we looked at the possible origins of these residues, looked at the leaf applications, or you could also call it infection due groundwater or other events of contamination. And here the phosphonate origin was proven, and exclude them for the wine growers. We did this. We were able to prove this. Of course, there are other sources of influx. This is just one step to prove the origin. And to reduce phosphonate, we carried out a container test, which is only carried out in the model. We will have subsequent tests in the vineyard, but this is what we already know. We have just confirmed what's already known, that BBCH 68 can reduce the residues, and if you use this for longer, you have higher residues, and all of that can also be seen in the vineyard. And I would like to thank everyone involved, because research is always teamwork. Thank you. I know, and this is my poster about the influence of the microbiome by applying compost tea. This is about the microbiome on the surface of leaves and grapes. Compost tea is made by using a small amount of compost and allowing it to be in water, ferment in water, adding oxygen all the time for about 24 hours. And that is all. The residues are enriched, particularly fungi and bacteria. Then we filter the liquid and apply it directly to the canopy in order to get the microorganisms onto the leaves and the grapes to work against infestations and disease. And we have seen some excellent efficiencies in viticulture, but there are fluctuations due to various weather conditions or application conditions or different compost types. The practical research network, VITIFIT, came up with a question whether we could maybe look at this in with a more systematic way to identify the efficiency and make it applicable in a more stable way. So we are carrying out examinations over 3 years to see how to look at the microbial composition of the compost tea and to find out which of those microorganisms establish themselves on the leaves and the grapes, at least temporarily. We also want to look at which of these microorganisms may have an antagonist effect against pathogens, such in particular the downy mildew. We found that even though it's difficult to read here, that there is a systematic shift of the microbiome on the grape and the leaves, particularly when it comes to bacteria. This was for 2023. Approximately 5% of the detected microorganisms on the surface of the leaves and the treated parts of the plant could be attributed to compost tea. We were also able to see that some of those microorganisms, mainly bacteria, had an antagonistic effect against various pathogens. The results from 3 years are going to be compiled soon. They will be based on 12 independent treatments in order to see which microorganisms can have an effect against these specific pathogens of the vine. Thank you very much for your attention.
Hallo zusammen, Paul Besrukow von der Hochschule in Geisenheim. Sie wissen ja alle, wie Weinbau funktioniert.
Paul Besrukow of Geisenheim University. So you all know how it works. We have a vineyard, we spray pesticides, and then we make wine. But as a matter of fact, we spray pesticides to protect the plants, but the plant also has its own tool to protect itself. They're not always sufficient, but they exist. And that makes them, and they actually produce their own chemicals, phytoalexins, and or stilbenes. And these stilbenes, for example, can be found in the root trunk. And we always cut these in December. And so our idea is to use this to extract the good stilbenes and to give them back to the plant in the following year. And but there are some challenges. 3 of them are shown here. One of them is what's the stilbene content and how is it influenced by different factors such as the variety, environmental factors, and the location. All of these play a major role on the quality of the construct. And then the second question is, how stable are these abstracts once they've been applied, when the sun shines on them, for example? We examined this, and we created new formulations that protect stilbenes against sunlight so that they can actually take effect. And the third point in the bottom left-hand corners, how first of all, we did a greenhouse experiment, and we saw that their effect is not just against copper. Blue is copper here, and green is stilbenes. But if you mix them, there is a certain amount of synergy, which means that they have a stronger effect together than individually. So, of course, all of this works in the greenhouse. So we then took this to the field test for over 2 years, and shown here at the top. And this showed similar results. Here, again, we had the grape cane extract, and it has a similar effect to copper. We were not able to prove the synergy over the 2 years. And finally, we looked at what happens with the grapes and the must that we get out, whether it's acceptable. And there's actually no change. And the only change is the copper content in the must. which is higher when you use copper, but that was to be expected. So that was our approach. And if you would like to find out more details, come and see us and take a closer look at the poster. Thank you very much.
Ja, guten Tag. Mein Name ist Stefan Schwab von der.
I'm Stefan Schwab of the University of Erlangen, Nürnberg, and my poster is on the production and characterization of solid lipid microcapsules, the CuCaps that contain copper. Tomorrow morning, I'm going to give a presentation on CuCaps. That's why I'm not going to go into detail about them now. I'm just going to talk about the production process of CuCaps. So plant protection nowadays don't just contain the active ingredient, but all kinds of ingredients. That is the formulation. And one of the formulations that we have developed are these solid lipid microcapsules. In order to make this kind of formulation sustainable, it has to be made from sustainable materials, and that is why we need various plant fats and waxes and integrate them into the microcapsules. And of course, also the production process has to be sustainable. That is why we've developed spray cooling. You may have heard of spray drying, which is a widespread process in industry for the production of small capsules. But that process is very energy intensive, requiring temperatures of up to 150 degrees. And energy efficiency is an important thing, and that is why spray cooling just works with between around 50 or just above 50 degrees. So this makes it more cost efficient and energy efficient. And here the process, as you can see, consists of 3 steps. The first is the mixing of all of the ingredients to create a melted product, which is then taken to a spray tower where it is sprayed through a nozzle, and the driblets then solidify to form the lipid microcapsules or particles that are then separated by the airflow to produce the final product. It is a simple process in 3 steps, so it is easy to scale it. This is important for industry in order to avoid having to need new technical components. A lot of the drying can be taken from other drying applications. And in Erlangen, we have a laboratory set up that can produce 30 tons per year. Then a product has to be analyzed and characterized after production. These are particles, a kind of powder. And so, of course, the first question is how will it behave? For example, there is dry quartz sand, which is but there are some products that may stick a little and stick to the plant. That's something that you may want. But we also don't want the powder to solidify into one big mass during storing. So you have to find basically the happy medium here. And of course, the whole thing has to be applicable. So because if you have a good technology that can't be applied, then that's no good. And that is why there was some criticism at the start. We heard that the filters were blocked by the product, and we had engineers, and that is why we built a field spray nozzle, and so we have a test bed for that now, where we test it. and where we can simulate the application process in order to ensure that our microparticles are applicable. And that is, of course, an important precondition. And then we work with cooperation partners such as Geisenheim University to check the effectiveness, the adhesion on the leaves and on the grapes. But I'll talk about that some more in my presentation tomorrow. This poster is about the production and the characterization of the solid lipid microparticles. So I'll be happy to answer your questions either near my poster or maybe we can discuss it tomorrow after my presentation. Thank you very much.